Hi, everyone. My name is Pranam. As Jennifer mentioned, I'm a professor of biomedical engineering and computer science at Duke University. And today I'm going to share with you the work that my team has done to develop peptides with generative language models. So a lot of this work actually began because of COVID, and we were really inspired by the work done to develop vaccines as well as monoclonal antibodies, which essentially can bind to viral particles outside of the cell and prevent them from entering. But what if SARS-CoV-2 enters the cell? How are you going to prevent it from replicating and infecting further? To solve this question, we looked at how SARS-CoV-2 interacts with the human ACE2 protein via which it enters. And by focusing on the crystal structure, we were able to use ACE2, cut it up into peptide sequences, and then through an iterative refinement process, find those that had high affinity or predicted high affinity to the RBD of SARS-CoV-2. But instead of just using this peptide as a binder, we actually took a human E3 ubiquitin ligase and we replaced its natural substrate specificity with our peptide, allowing us to now bind and ubiquitinate viral proteins, leading to its proteasomal degradation. And we showed that when we put this peptide fusion thing into viral cultures, we could reduce the infectivity competency of the virus by over 60%. So that was pretty cool stuff. But I think what was even cooler was this idea of what we call ubiquibodies, where if you can design a peptide to any target protein, you can degrade it. Because the goal of our lab is to create a CRISPR analogous system for targeted protein degradation. Hopefully you guys know CRISPR. It's really easy. You just have a target DNA sequence. You can design a guide RNA that goes there, attach it to a Cas9 enzyme, bind and edit, right? That's why CRISPR is so cool. But what if you could do the same for proteins? What if you could take a target protein sequence, design a peptide, attach it to an E3 ligase, and now bind and degrade your protein? How do we achieve this level of programmability? Because a lot of the proteins you want to degrade are pretty undruggable. You don't have small molecules that can bind to there's no binding pockets on these proteins, and a lot of these proteins are also super disordered, so you can't use structure-based methods. But I think we can solve this question because we have NLP. And you've heard about it a lot. These high-parameter language models can capture really important characteristics of language: syntax, semantics, grammar, to name a few. But protein language models, as you've heard, have also been trained not to capture syntax, semantics, and grammar, but to capture structure, function, or interaction. And you've seen this go generative, right? You may have heard of DALL-E, where you can put a text of a corgi playing a flame-throwing trumpet, and you get a picture of a corgi playing a flame-throwing trumpet. And the way that DALL-E works is by training this contrastive latent space where text and image embeddings that are close together that are associated are close together. So my student Kalyan was like, okay, instead of text and images, what about peptides and proteins? And so he took the putative peptide protein data set and trained a joint encoder system where, using ESM language model from Facebook, we were able to identify peptides and proteins that do bind being close together in the latent space, and those that don't bind are far away. So we use that as the objective. And to actually generate peptides, though, to push through this CLIP module, we use protein-protein interactions from comprehensive data sets that we curated, took the known interacting proteins of the target and just cut them up and threw them through CLIP to identify the peptides that had high selectivity to our targets. And we showed that when our model, we were able to rank our top peptides from the CLIP model and put them into the ubiquibody architecture. In the lab, we were able to degrade various undruggable proteins like KRAS, for example. But can we do better than just randomly cutting the target or the interacting partner? Garrick was like, yeah, I think we can, because we now have all these interacting protein sequences. And what he did was train and fine-tune ESM2, the language model, to now be able to predict the per amino acid interaction probability. We collected a huge data set, about 450,000 peptide protein interactions that we derived to identify interaction probabilities, and fine-tuned ESM2 with an MLP head to now predict the cross-entropy loss, the interaction probability of each amino acid, such that you can now get a landscape of the interaction probabilities across the interacting partner and just cut the parts that have the highest interaction probabilities to get your interacting peptides. And so And so now, so this we called salt and pepper for peptide prioritization. And salt and pepper identifies interacting hotspots on partner sequences very accurately, and also does as well as structure-based methods that also identify structure hotspots on structured co-complexes. We showed that those peptides, if we incorporate them into the ubiquitobody architecture, degrades beta-catenin in colorectal cancer cells, leading to reduction in tumorigenic signaling in those cells. And we show that this happens because these peptides bind with super high affinity, low nanomolar affinity. For peptides, it's pretty good. And we also show in mass spec that these peptides have very low off-target rates. We only degrade beta-catenin, as you can see. So take a read. I know I'm going quick, but there's one more thing I want to tell you. But there's another, this paper's on the preprint, the code's available. You can use salt and pepper. But remember, salt and pepper and cut and clip use partner sequences as scaffolds. Can we do this purely de novo? That's been the question we've tried to answer. And so I think we have to go back to clip to answer this question. Instead of cuts, what if we could use realistic de novo peptide sequences, right? It's things that we just want to screen. However, there's about 10 to the 23rd possible 20-mer peptides. You can't screen them feasibly. Maybe you could, but that's not a good idea. So we wanted to answer the question, how do you make a ton of naturalistic peptides? So Saha said, hey, let's take ESM2. ESM2 has this very expressive latent space, and we can put real peptides and get their embeddings. Instead, now we take those embeddings and we perturb them with Gaussian perturbations to get different embeddings that still meet the ESM2 criteria, and then decode those back into naturalistic peptide sequences, which we then throw through a clip model that can rank the specificity of those peptides. So we call this pepperclip, and pepperclip can generate very naturalistic peptides. You can adjust the variant scaling factor on the Gaussian perturbations to get good peptides that you'd like. And then for the clip module, we actually improve this module first by training on this noisy data set of both weak and strong PPI interactions, but then testing them only on strong peptide protein interactions that have very specific criteria. And we show that this does really well. There's a top 10% accuracy of about 92%, meaning in the top 10% of peptides that you test, you're going to get a good peptide in that set. And you can see that the peptides and proteins that do match have high clip scores. Peptides and proteins that don't match, don't bind together, have lower clip scores. So we benchmarked this with AlphaFold multimer, co-folded the peptides that we generated with a target. We show that our peptides interact just as stably with target proteins as real peptides that interact with that protein. We did that for a few different targets. You can see the ones we generated on the left. And we show that when we integrate these peptides into our E3 ligase architecture, we can degrade targets like TRIM8, which is an undruggable target that causes Ewing sarcoma. We put those into Ewing sarcoma cells, and we induced apoptosis in those cells. So we're killing those cancer cells. Take it out for a spin. The code's available. It's really easy to use in a Colab notebook. Last thing I want to say is that you can see that language models are clearly good unconditional generators. What about target conditioned peptide generation? So Leo in the lab has trained this really cool model, not GPT. We have trained GPT, it doesn't work that well, but here we've trained mass language modeling where we just mask out the entire peptide sequence given a target, asking the model to, via cross-entropy loss, to just generate a peptide given a target sequence, just unmask it together. These produce peptides with low perplexity, so these are really good peptides. I'll be done in 1 second. And these peptides have very stable binding. Last thing, these peptides are used for degradation. We've used them for other things like stabilization. We've stabilized proteins like beta-catenin, FOXP3. So overall, instead of programmable RNA-guided gene editing, we're doing programmable peptide-guided proteome editing. So thank you so much. And we have great collaborators, great funders, and thank you to you guys for listening to my talk.
We have time for a 30-second question. Any takers online? So are you doing the computational and the wet lab work?
Yeah, so our lab, as you can see, we have a full computational team. They don't know how to do experiments. They don't probably understand biology. And these guys don't know how to do computation. They just work together. And we do both validation and like cool.
You're the only one that understands both?
Yeah. But maybe
by design?
Yeah, by design. I'm hoping that one day I'll get a student who can do both. But you know, it's hard. It's a lot of work to train on both.
Yeah. All right. Okay, great.
Yeah, thanks, guys.
Thank you.